Hello, my name is Ben Shaberman, and I'm a science communications advisor at Hope and Focus. And I'm pleased to present this general overview of Leber congenital amaurosis. I'll be covering what LCA is, how it affects the retina, the genetics behind LCA, and at the end of this talk, I'll provide a quick overview of some of the emerging therapies for LCA. If you're interested in more information about LCA, we encourage you to go to hopeandfocus.org, or you can send an email to info@hopeandfocus.org. So LCA is really a family of rare inherited retinal conditions. And I think that's a really important concept to understand from the beginning, is that it's a family of conditions. There are many different forms of LCA based on the genetics that an individual has. And we'll be talking more about that throughout the presentation. The genetics, the mutated genes affect the retina, and the common theme for people with LCA is that they have significant vision loss at birth. And usually that vision loss is progressive. LCA affects as many as 250,000 people around the world. In the US, experts believe between 8,000 and 10,000 people are affected. Now, LCA is what we call a monogenic condition. That is, each person that's affected has one mutated gene that's causing the disease. There's one gene that's the culprit. The challenge with LCA is it could be one of at least 27 genes that's causing the disease. And again, we'll be talking more about that. Now, LCA is in some ways similar to other inherited retinal diseases like retinitis pigmentosa, Stargardt disease, and Usher syndrome. These conditions are also monogenic and they're also inherited. Generally speaking, they're not quite as severe as LCA. And in fact, sometimes LCA can be confused with retinitis pigmentosa and vice versa. Now, on the right side of this slide, I have an image of an eye, of an eyeball, a side view. And at the very back of the eye, on the inner part of the eye, in red, is the retina. The retina lines the back of the eye. It's a very thin piece of neural tissue, only about a half millimeter in width, lining the back of the eye. Now, the retina is connected to the brain ultimately through the optic nerve and some other axons, but the information that the retina captures travels across the optic nerve to the brain. And to the right of the image of the eyeball is that half millimeter retinal tissue magnified considerably. And let's talk more about what the retina is. So the retina, in simple terms, operates like film or digital sensors in a camera to capture light, to capture the images that we see. And what the retina does is it converts that light into electrical information that again is sent over the optic nerve to the brain. And the brain uses that information to ultimately create the final images that we see. So the retina and the brain have a really important partnership for vision. Now, the cells that are most important for enabling us to see are the photoreceptors, or the rods and cones. Those are the two types of photoreceptors we have. And the typical human retina has about 125 million photoreceptors. And you can think of the photoreceptors as working like antennae to capture light. And the photoreceptors begin the process of converting that light into electrical information. And we have two different types of photoreceptors, rods and cones. Rods give us night vision, and the vision and the ability to perceive things on our periphery, and rods are more concentrated on our peripheral retina. Now, cones are concentrated more in the center of our retina, and cones give us the ability to perceive detail, to recognize faces, and to read. Cones also give us color vision and the ability to see things in lighted condition. And on this diagram, I have rods and cones circled. They're the longer cells toward the top of the diagram. And again, each retina has remarkably 125 million photoreceptors or rods and cones. So let's review quickly the genetics of LCA. And as I've mentioned already, there are at least 27 genes, each of which, when mutated, can cause LCA. That's what gives the family of LCA forms its diversity. Each mutated gene affects the retina in a different way. Some mutations affect the development of our photoreceptors and rods and cones. Some affect the health and survival of rods and cones. Others affect the function. And excitingly and importantly, genetic testing is now widely available and accessible so that a person or a family can determine which mutated gene is the culprit. And you can learn more about genetic testing on the Hope and Focus website because it is important to identify that mutated gene. Now, I want to give you a quick overview of what genes actually do. And you can think of genes as like recipes. And our cells read the genetic information or these recipes to make proteins. And ultimately, it's the proteins that are so important for the development, health, and function of the retina. And when there's a misspelling in a gene or a misspelling in these genetic recipes, bad things can occur. There can be an incorrect protein or insufficient protein. And that's what leads to diseases like LCA. So LCA is usually, not in all cases, but a vast majority of LCA cases, the condition is inherited recessively. And what that means is parents are unaffected carriers, their vision is fine, and they have no idea that they're carriers of an LCA gene. And what can happen when a mother and a father or two people get together and they both happen in these rare cases to be carriers of the same LCA gene, each of their children has a 1 in 4 chance of inheriting both of those mutated copies, one from each parent, and then actually getting the disease. So in just about every case of LCA that occurs in a family, at least with the first child, LCA comes as a surprise because parents have no idea that they are carriers of an LCA gene. And on this slide, I list the 27 known and validated LCA genes. And the different gene names are in parens, like GUCY2D, RPE65, AIPL1, etc. And sometimes we refer to each form of LCA by the gene name, but experts also refer to the different LCA forms with an acronym with a numeral after it. So sometimes they'll refer to GUCY2D forms of LCA as LCA1 or RPE65 as LCA2 and so on. So to finish out, I wanted to, at a very high level, talk about the different therapeutic approaches for LCA. By far and away, genetic therapies are the most common therapies in clinical development for different forms of LCA. And when I say genetic therapies, they usually involve delivering new healthy copies of genes to replace or augment the mutated copies, but they could also involve in some way correcting the mutated gene, doing more of a cut and paste. Excitingly, we have an FDA approved gene therapy for at least one form of LCA, LCA2, which is caused by mutations in RPE65. That gene therapy is called Luxturna. It was the first FDA approved gene therapy for any eye condition or really any inherited condition. And we're excited that it happened to be for LCA2. This gene therapy has restored significant vision to children and young adults who were, of course, born with LCA2. Some of these kids and young adults have been able to put away their navigational canes. Some could see the faces of their loved ones for the first time. Some even saw stars in the sky after getting Luxturna gene therapy. And we're excited that some other gene therapies for LCA are also showing very good results in clinical trials and restoring some vision. Now, another approach to treating LCA could be what we call neuroprotective therapies. These aren't targeting the mutated gene. These are what we call gene agnostic or gene independent approaches. And the idea behind a neuroprotective therapy is that you're just trying to preserve photoreceptors, to preserve the overall general health of the retina, regardless of the mutated gene causing the disease. Now, neuroprotective therapies could be drugs, let's say oral drugs, or they could be gene therapies that aren't replacing or augmenting the gene, but doing something else to, again, just preserve the function and survival of existing photoreceptors. And I'll add that neuroprotective therapies are in clinical trials. Another approach that's in clinical trials and showing some early exciting results is optogenetics. And optogenetics is targeting people with advanced vision loss, and it's for people who have lost most or all of their rods and cones. And what optogenetics does is it enables other cells in the retina, like bipolar cells or ganglion cells, to sense light, in essence, work like a backup system for photoreceptors. Bipolar cells and ganglion cells, they have important functions in the retina naturally, but they don't naturally process light. What optogenetics does, it bestows light sensitivity, the light sensitive function to bipolar and ganglion cells. And again, in clinical trials, we're seeing, especially for retinitis pigmentosa, that optogenetics can restore some meaningful vision. And we think it has potential to help people with LCA. Cell-based therapies are not quite as far along in clinical trials as the other approaches that I've mentioned, but they are starting to move into clinical trials. And the basic idea behind a cell therapy is that you're replacing lost photoreceptors. You're putting in new rods and cones to replace those that have been lost, to potentially restore uh, some meaningful vision. And as I've already mentioned, some therapies for other retinal diseases, especially RP, could apply to people with LCA. So it's important that we keep our attention on these other emerging RP therapies, especially neuroprotection and optogenetics. And again, there are many exciting clinical trials underway for these therapeutic approaches. So that concludes my overview of LCA. Again, if you want to learn more information, go to hopeandfocus.org or feel free to send us an email at info@hopeandfocus.org. Thanks.